The overall goal of this method is to investigate changes in the lipid composition of neutrophils during the release of neutrophil extracellular traps by using thin-layer chromatography and HPLC.
This method can be used to gain a more detailed information about the mechanisms that are involved in the formation of neutrophil extracellular traps, or shortly named NET formation.
The main advantage of this procedure is that it provides information on the role of lipids in the general function of neutrophils by utilizing an easy-to-follow protocol.
The isolation of human blood-derived cells will be demonstrated by a former PhD student of our lab, Ariane Neumann, and she will be assisted by Friederike Reuner.
The preparation of cells for lipid analysis will be demonstrated by Brogden, a postdoc in my laboratory, and Diab Husein, a master student in the biochemistry program.
To isolate human blood-derived neutrophils, layer approximately 20 milliliters of blood onto 20 milliliters of sodium ditrizoate dextrin solution near a flame without mixing. Centrifuge for 30 minutes at 470 times G without breaking.
It is critical not to mix the blood phase with the density gradient solution. After the centrifugation step, plasma and mononuclear cells need to be carefully removed to avoid a contamination or unspecific activation of cells.
Remove the mononuclear cells and the yellowish plasma.